Hello, my name is Advaith Dinesh, and I'm a rising senior at Northwood High School in Irvine, California. My great mentor for this project was Elliot Garling, and with her support, I was able to complete this research paper. Now, to start off, why did I choose this specific topic? Well, a few years ago, my mother shared that my grandmother passed away due to lung-related complications. And since that moment, I've been kind of hooked on researching more about diseases or problems of the lung. Therefore, I started to look at more magazines and articles related to that certain topic. And in my science classes, I started to learn a bit more about cancer. So when it was time to finally choose a topic, I decided to go with lung cancer because I was really interested in what I learned from school. Now, to start off here, a bit of an introduction. Lung cancer kills more than 100,000 individuals in the United States each year. Yet conventional treatments, or treatments that we know of the most, especially chemotherapy, lack specificity and kill healthy and cancerous cells alike. Now, for this paper, I decided to focus on non-small cell lung cancer, or NSCLC, as it is the most common type of cancer, accounting for 81% of all lung cancer diagnoses, where abnormal cells form um, in lung tissue and multiply. Now, cytotoxic, which also means toxic to cells. So, cytotoxic agents-based chemotherapy has been the main approach for the treatment of a wide range of cancers for decades, really. Now, most chemotherapy drugs have a low therapeutic index, or in other words, the range between a therapeutic and a toxic dosage is pretty small. Now, you know, significant side effects are frequently attributed due to the lack of selectivity or targeting capability to an intended molecule or cell surface receptor, I mean. Now, moving into target therapies, for a little bit of history on this, during the beginning of the 20th century, scientist and Nobel Prize winner Paul Ehrlich developed the concept of a magic bullet, or basically a drug that would travel straight to its intended cell structure target. Now, over 100 years of research and therapy have led to the formation of novel cancer therapy that's essentially built on the idea of the magic bullet. And really, the main idea here is that targeted therapy drugs for cancer means more precision. And more precision leads to fewer side effects because healthy cells aren't going to be, destroyed. However, in order to understand ADCs or antibody drug conjugates a bit better, we need to have decent knowledge of antibodies. Now, antibodies are the immune system's weapon for combating infections, cancer, and other disorders, and they fight by engaging with unwanted particles, proteins, and receptors, which we commonly refer to as antigens. Now, this IgG antibody or immunoglobulin antibody shown right here on the right um, consists of two heavy and two light chains. And then we have variable regions near the top, constant regions on the bottom half. It's sort of said in the name, but variable regions are able to change shape depending on their target antigen. Now, the antigen binding site you can see is located on the top half as well. Um, so now if we move on to the structure of an antibody, um, antibody drug conjugate um, consists of an IgG antibody that supports chemical linkers on the side, acting as branches for the cytotoxic drug to latch onto. So you can see in the image here, that's our antibody, got the cytotoxic drug, and then the linkers that attach the drug to the antibody. Now, primitive mechanism of action. ADCs are administered intravenously or through the IV in order to degrade um, degradation due to digestive enzymes or other things like that. Now, once they're in circulation throughout the bloodstream, ADCs will then locate and locate their target antigen and bind. And from there, the ADC becomes ingested or taken inside the cell via a process called endocytosis. Now, it's basically in this process, the ADC is encased in a bubble-like structure made of membrane slash phospholipids, and this structure is we call an early endosome. Now, the early endosome will then fuse with the fuse with the lysosome, um, or lysosome is also an organelle of the cell which contains certain enzymes that can degrade substances. However, if a drug is attached to a cleavable linker, remember the linker is a thing that attaches the drug to the antibody. So if that linker is a cleavable linker, the ADC does not need to interact with the lysosome to release the drug. But essentially, once the drug is free from the ADC, it can then travel to the interior of the cell and start to induce damage upon the DNA or other structural components. You can imagine if uh, the drug, stops a process or stops a certain component or structure of the replication cycle or of the DNA, that means the cell cannot keep replicating, and replicating cells is what causes cancer or an excess of replication. So if a drug stops that, then cancer can be stopped. Now, as the target dies, some of the cytotoxic drug can leak out into neighboring cells, which most likely be cancer cells, and this further multiplies that damage. And we call this the bystander effect. Now, a bit more into current research. I'm going to talk about clinical trials and how that sort of works right now. So, in general, clinical trials are conducted in four stages. Um, a limited group of people are tested for the first time in phase one trials. It's just to find out about the safety and potential negative effects. After phase one, phase two trials involve around 100 to 300 participants, which is a larger population. And phase three is from around 1 to 4 years, examining a significantly larger sample size, 1,000 to 3,000 people, and just verifies efficacy, tracks adverse events, and just comparing the treatment to other approaches. Now, phase four trials continue long after the medication is licensed and made available to the public, and it covers a wide range of areas. Once again, now, looking at some more specific examples of ADCs that are being used to treat, cancer. One of them is uh, trastuzumab deruxtecan, also known as Enhertu, and it's been conventionally used to treat breast cancer, but it's also receiving more research in the treatment of NSCLC. Now, Enhertu is an ADC that targets human epidermal growth factor receptor 2, also known as HER2. And it's a key cell surface receptor, that protein that's involved in healthy cellular growth and division. However, the problem is in cancer, HER2 is often mutated and expressed on the surface of malignant cells. Now, in a phase one trial evaluating HER2 targeted ADCs in NSCLC patients, researchers found that approximately 59% of controlled patients reacted effectively to the treatment, with 88 demonstrating disease control or the ability to suppress tumor growth. And more of these trials are happening, so a lot of exciting research is on the way. Now, um, cytotoxic payload produced by ADCs for lung cancer has been shown to have tolerable safety profiles, which, the reported side effects falling within a known or better range. And the really exciting thing is that this means that patients can manage NSCLC ADCs without necessarily experiencing the same crippling side effects that other treatment modalities might cause. I mean, think for chemotherapy, you can I'm sure you've seen or have had people in your family who have had to undergo chemotherapy, and the process is not very it's not great. So having a treatment like ADCs can help mitigate that response. Now, uh, some limitations. We have to talk about limited antigens. So limited antigen targets pose a pretty huge problem for developing more ADCs. Um, Identifying specific receptors glycoproteins or other markers that are highly expressive on tumors, but normal uh, normal when it comes to you know but like minimally expressed when it comes to normal tissues can be pretty difficult. So even though ADCs and moving on to the next limitation, even though ADCs have comparatively fewer adverse effects than chemotherapy, for example, some off-target toxicity still exists. It's not completely removed, so patients still might have certain side effects, but once again, it might not be as bad, or it isn't as bad as something like chemotherapy. Now, some ethical considerations that have to be looked at is cost and affordability. Patients often seek better treatment options for their cancer, but unfortunately, this can be swiftly stunted simply due to the cost of certain therapeutics. If we look at, Casgevy, for example, this is a therapeutic from Vertex Pharmaceuticals and CRISPR Therapeutics, and it's the first medicine to be approved in the U.S. that uses CRISPR, or which is a gene editing tool, and this can help a lot of patients with sickle cell disease. Uh, the really striking thing, though, is it costs $2 to $3 million per treatment, and that's just completely out of the range for such a wide majority of people. Um, so really, affordability ties so closely when it comes to treatment, and these high costs create detrimental barriers to those without proper insurance or overall wealth. So ensuring this equity and equal access can really alleviate the stress of inducing an unhealthy financial burden that would be usually placed upon people. Now, a large subset of ethical considerations should be put on the experimentation of animals um, when in vitro options like phage display exist. Many find it questionable to utilize the bodies of animals who cannot consent to research. And as mentioned, antibody testing, which uses immunization, can cause suffering for animals. And when we look at gender, women tend to have um, usually or comparatively, in comparison to men, they have usually more adverse effects to treatments, and not enough. There's not enough equity or equalness when it comes to researching effects and treatment on women as compared to men. So there has to be better awareness and better standards when it comes to research and making sure that both genders are equally looked at and tested for. Now, for my conclusions and final directions, um, let's start off with this. One of the most popular forms of treatment, which we know as chemotherapy, is acknowledged for fighting certain cancers like lymphoma. However, in the fight against other cancers, chemotherapy often does not cure, but it rather just increases the longevity of life. It does not care, it just increases longevity. Often it's given as a palliative measure, not necessarily a cure, and it's often observed to have daunting side effects. Um, where other therapeutics may lack, ADCs may offer advantageous benefits. And like I've said throughout the presentation, one of these advantages is the targeting ability of ADCs. Now, as mentioned, we have to consider the high cost of treatment. It remains as a large barrier to equal access to life-saving medical care. Additionally, not all treatments are guaranteed success. A certain number will fail in production, and subsequently their costs are added onto later successful therapeutics to remedy their profits. And so the ability for companies to patent new drugs patent new drugs, allows them to set higher prices on these ADCs because there's no real competition when it comes to the price range. Now, ensuring that the produced ADCs are representative of, uh, for a diverse set of individuals also remains a challenge. And depending on race, gender, effectiveness, or depending on race, gender, and other factors, the effectiveness of ADCs may really vary. Now, in conclusion, ADCs are a promising candidate for cancer therapy and have really an enormous potential for treatments against NSCLC and other cancers. As the search for better ADCs continues, a variety of questions must be asked to procure the most effective and non-harmful ADC that can be distributed to all patients in an equitable manner. Thank you.